We will then proceed with the second talk. It's going to be by Enrique Sapena Ventura. And Enrique has a background in biology with a master's in virology, but also in bioinformatics. And he's a data wrangler for the HCA Data Coordination Platform, the DCP, at the European Bioinformatics Institute. So his job consists on ensuring that the data flow is as efficiently and easy as possible from the scientific community to the DCP. So I believe we can all understand how important his job is. And let's welcome then Enrique with his talk.
Good morning. Today, we are going to provide with an overview of the HCA's data coordination platform, what it is, what we do, and how you can contribute. But before we dive into the platform, let's give it a little background. So, the Human Cell Atlas is a very diverse community, composed of specialized researchers from very different backgrounds around the world, but together profiling millions of cells across different organs of study, techniques, and data formats. And this body of data is just growing larger every day. So, overall, the HCA community is about as complex as the cells they are sequencing and comprised of many types of researchers. This community includes experimental researchers, who are the ones performing the experiments, isolating the cells and sequencing them, computational biologists, identifying how to best approach and analyze sequencing data, software engineers, and tool developers who create new ways to work with data, and of course, every type of researcher in between. So, each of these community members face different challenges to accomplish their goals. For example, an experimental researcher might need to contribute, analyze, or visualize data, whereas a computational researcher might need access to the data to use it for analysis. And engineers, on the other side, might need to download bulk data to test their tools that will better serve our community. So, with all this diversity in mind, with data, tools, research teams, and community members, how do we bring everything and everyone together to form a meaningful atlas? In order to do this, we need a public platform that is comprehensive, including many data types to describe human cells, inclusive. So, with many labs working together on different tissues, we want the platform to minimize sampling bias as well as remove barriers of entry to both contribution and access. We want it to be organized so that data is centralized and standardized. It also needs to be accessible to researchers worldwide. And of course, it should be dynamic, with a fast response to new data types, techniques, and analysis methods. As you know, this is an evolving field. So these are very high-level aims, but what must this platform do to accomplish them? First, it needs to bring all the data together. We must ensure that the diverse consortia data is harmonized and described in a usable way with standardized metadata, so we can enable consistent management of data and therefore grant access to raw, process, and metadata. So, although the community generates and analyzes data for very different research questions, we still need a way to combine the data for building a common atlas. We need a way to make sense of the data. The standardized processing pipelines have to allow us to use all this data by providing with standardized outputs while still being responsive to the community needs and usable by any researcher. In addition to having standardized processing, scientists need ways to contribute and process data on their own timeline. The platform must be able to handle this evolving data by providing regular releases of consistently processed data. Usability of this version data is key to accelerating science, but it's only possible when integrated with your day-to-day research. So this open platform has to integrate with the tools, portals, and software that fit independent research needs the most. But so an open system is only half the battle. We must make the systems work together by creating standards for the community that they can use to describe single-cell experiments, standards that will allow developers to build tools against the data to harness its full potential. So now you know what a platform should do, and that's the vision that we have for the HCA Data Coordination Platform. All the components you can see in this slide make the DCP a scalable platform led by a very diverse set of teams, open to individual researcher input, but proudly coordinated with other communities, where you can organize and interrogate single-cell data. So let's learn a little bit more about how it actually works. On the next set of slides, I will unpack the flow of data through the DCP, from the very first point, which is contribution, to analysis and data access. Let's break this down a little bit more, starting with contribution. The flow of contribution begins when researchers contact the data wrangler team. Once the suitability of their dataset has been assessed and we are sure it meets the legal requirements, we will begin an iterative process to curate the metadata to a high standard. And once the data is uploaded, we validate it for the researcher and archive it, becoming publicly accessible, getting accessions for their manuscripts, and becoming part of the next DCP data snapshot. Once a submission is ready, it is handled to the data processing and pipelines team, where the data and metadata is processed with standardized pipelines that produce alignments, gene matrices, and quality control metrics. These pipelines are cloud native, documented, and publicly accessible. That meaning that you can try the pipelines on your own in Terra, either on your 10x dataset with our Optimus pipeline 3'N, or on your SmartSeq2 dataset. Otherwise, if you are interested in accessing and exploring the raw or process data that is already available, you can do this from our data portal. On the data portal, you can find all the data that has been brokered into the DCP, being able to filter the datasets by their metadata, for example, by the age of the donors, and select the projects of your choice, creating cross-project cohorts. These cohorts can either be downloaded locally or exported to cloud-based tools that support standard handoff mechanisms, making it easier for developers to extract data automatically. We currently support handoff to cloud-based platforms like Terra, Single Cell Portal, and UCSC Cell Browser. You can also find and access community tools on the data portal, where you will find a great collection of community portals and tools for the most up-to-date analysis, visualization resources, and many others. Feel free to visit data.humancellatlas.org/analyze to discover it for yourself. And any researcher and developer of the community is welcome, more than welcome, to contribute with their own tools. So now that you know the data flow and what you can do with the DCP, we would like to highlight some upcoming features. In the next iterations, we would like to provide contributor-generated analysis files, which will allow researchers to share their own analysis, batch corrected and normalized when possible, in order to add the deep knowledge that each researcher has about their own tissue to the standard processing we provide with. We are also working towards allowing managed access datasets, given that some human data is delicate and researchers should have the ability to share their research to the greater community. We are going to have a pilot, so if you have any managed access data and you are interested in joining, please feel free to reach out. whether you're interested in joining the pilot, contributing with your data, interrogating the data that it's already there, or just providing feedback, we encourage you to reach out. We are an evolving platform and all the feedback and questions are very welcome. So please reach out on data-help@humansatlas.org. Lastly, if you would like to contribute with your data, please email our Wrangler team with the email in the slide, and we will be more than happy to start with the submission process. But before we end today, we need to say a huge thank you to our sponsors and team members who make the DCP possible. A special shout out to CCI, as without their funding, this project would not have been possible. And another shout out to all the people who work very hard in all the teams. Without their dedication and effort, this wouldn't be possible either. Also, a huge thank you to all our funding partnerships and partner consortia. And thank you for listening today. Any questions?
Enrique, thank you so much for your presentation. Yeah, I think it was so complete that the majority of the questions here, if not all the questions that appeared here from our participants, were answered by your presentation. There were many technical questions about how to use the platform, how to share data, how to use data, and I think you managed to answer all those questions. And also leaving it open for people to reach out to you and possibly ask specific questions in the near future. I think that was great too. And I think your presentation was also great to show how dense and how complicated it is for you guys to try to put all this information together. Maybe if you want to say a few words about that.
Oh yeah, of course. It is a great challenge to put this platform together. First of all, as I said in the video, you need to envision what the community can, well, not envision, you have to ask the community, but you have to come up with what the community needs and how to accomplish that, and then build it. Even if you have that idea in mind, it's not an easy platform to build, and there are many teams, many institutions working on this, and yeah. Also,
All of them with different ideas of how they want it to look.
Of course, yes. Yeah, and also I forgot in the video to mention. I would like to also thank to all the organizers of the HCA Latin America. Completely forgot, but really thank you for this opportunity.
Thank you, Enrique, so much. It was really great to hear from you. We have to move on because we are really running late in time. Thank you.